Good afternoon. Thanks for the invitation for joining this HPC summit and to share with you some of the study we've been working over the past decade. This is the outline of my talk. So I'm going to go through all these different topics in the next 15 minutes. So I just want to share you the traditional methods. When you do the biomedical research, you're going to spend a lot of time doing experiments, but generate a very small amount of data. But with this, the invention of the high throughput analysis, actually the experimental time is very, very short. However, it creates lots of big data. That's why this high throughput analysis actually revolutionized in the biomedical research. So there are many different biobanks across the world and regions. You can see here I listed include US, UK, and then even some in the Asia, including Taiwan Biobank. So a lot of countries and regions are putting great efforts to understand or combining different omics data into one place. So if you ask me what are the big data in biomedical research, I personally feel biomedical image is one of the big data. In terms of the vital sign, the physical signal, I don't feel it's a big data, even though you can collect those data continuously. The other one is the biobank data, from this is the example, Taiwan had 31 biobank database across different hospitals, and then also their medical records, and then Taiwan Cancer Registry database. So I personally strongly feel that I think the largest big data in biomedical research is next-generation sequencing data. Okay, as you can see, every time when you finish a NGS experiments, it will create probably 2 terabytes of data, and after many analysis as well, everybody is in the range of a gigabytes range. So it requires lots of computational resources, space, storage, and also computational time. So UK Biobank, I personally feel is probably the best biobank in the world. So they're collecting over 500,000 people, their genomics, and then their clinical data. So this is just show you an example, a paper published 3 years ago from UK Biobank. They combine genome-wide association study and then the brain imaging phenotype. So they identify a new biomarker related to brain. So this is a very good example showing that combine different data type, so you can have innovative research. So in the next couple of slides, and show you some of the research focus in my lab. So we actually dedicate a lot of time to develop a bioinformatics analytical tool. We have developed many different expert systems as well as R-based analytical tool or Python-based analytical tools. So I will introduce a couple of them in the next few slides. We also dedicate ourselves over the past few years on the AI deep learning, and then other personalized clinical medicine as well, metagenome, and then de novo sequencing as well. We actually is the first team in the world to do the whole genome assembly for microbial, which is the national bird of Taiwan. So this is one of our most successful expert system, I think, integrated genetic database. So it's an integrated system for characterizing enriched function and pathway of microRNA target. So we actually combine 9 different microRNA database, include those predict genes and their function into here. We also provide many different analytical tools here. So this is the screenshots of our MiDSystem, and then this is QR code. You can scan and go to this website directly. So ever since it was launched back in August 1st, 2012, over the past 9 years, we have over 86,000 visitors coming from 94 countries in the world. And then this paper was published in PLOS One, and then we now have 250 citations. So a database developed by a single professor in Taiwan can be well recognized by all the researchers in the world. That's its pretty big achievement. This is another database. We actually integrate cell line and clinical sample, their gene expression profile. So we combine cell lines and clinical sample, roughly around 5,000 microarray gene expression data. So we provide many different tools, and then we also have this user-friendly graphical display from our website. So it was published 3 years ago in database. And then you can see here, every year this database published more than 100 articles. We are the most read article from their websites. Ever since it was launched, again, we have over 27,000 visitors coming from 73 countries. We have 14 citations so far for this particular paper. This was another one published 2 years ago. It's combining genetic variants and then gene expression and also their clinical significance, so into one place. So we combine many different genetic variants coming from different ethnical groups. So, and then we have different species, human, mouse, and zebrafish. So, and over 700 million variants are included in this database. So this is a screenshot. So you can select whatever ethnical group, the genomic database you want to compare, and we provide different functions. For example, their allele frequency across different ethnical groups, and then their clinical significance, which particular genetic variant may be related to different diseases, also showing here. So this is a QR code. You can go to the website directly. So this is another analytical toolkit we just published earlier this month. So for mutation analysis in cancer genomics. So you can input the VCF data and go through, and we provide different analysis and visualization modules for cancer genomic studies. So you can see we provide 9 analysis and visualization modules. So you can do the mutational signature analysis. You can do mutational landscape analysis. You can also do, we include 3 analyses here. This one, HRD, stands for homologous recombination deficiency. Homologous recombination is one of the DNA repair mechanisms in our human cell. And also the chromosome instability and then whole genome doubling. In addition to that, you can also, based on their mutational signature, you can try to look for what are the actionable genes can serve as a biomarker, and then look for target therapy drug can be applied for specific patients. This is another toolkit. It's an R package. I think nowadays a lot of people are doing RNA sequencing analysis. However, it requires really quite extensive computer knowledge. So we developed this R package that would have been deposited into a bioconductor. So you can analyze your RNA-seq data with very minimum efforts. So we divide it into 6 different steps. Due to the time limit, I only have 15 minutes. I'm not going to go through all the detail, but you can download this package from Bioconductor, and then you can input your data and go through all the process. You even can compare, if you have 2 different groups, RNA-seq data, you can compare what are their gene expression level across different groups by using this R package. And it was published a couple years ago in IEEE ACM journal. Metagenome study has been it's one of the very hot field over the past 5 years, because the gut dysbiosis, the gut microbial imbalance actually create lots of problems, including cancer, nerves, cardiovascular, all kinds of different diseases actually are all related to your gut's microbiome. So that's why we also developed two different integrate analysis and database for studying metagenomics. So one of the popular to study bacteria is using NGS to sequencing the prokaryotic 16S RNA ribosomal RNA gene. So it was a relatively complicated process. So we combine everything into one place, and then it's a very user-friendly system. So you can upload your data and then go through our process. You can know the alpha diversity, beta diversity. You can even use this to study longitudinal analysis. For example, you are taking sample for one individual at different time points. So you will be able to see what are their gut microbiome changes across different time points here. Another level of study is using shotgun sequencing. So we also, this again, just published earlier this year. It's a MiDSystem. So it's a comprehensive online system for de novo assembly of microbiome genome. So you can do a shotgun sequencing of specific gut microbiomes, for example, the fecal samples. And then through this, we provide different analytical tools, and then we also provide some visualization, user-friendly display, so you can use this system to analyze your shotgun sequencing data. So we also established a database for Taiwan healthy individual, their gut microbiome. So this is the website. We provide analytical tool and for people to analyze their microbiome data as well, and also provide the baseline for particular study if you want to use. So as I mentioned earlier, we also dedicate ourselves into AI area, deep learning. So we just published a paper a few months ago in Briefing and Bioinformatics, which is a very high impact journal in computational biology. The impact factor is 11.6. So we apply this AI sequencing data pipeline as a new model to apply forensic identification and then application, and also for cancer classification applications using whole exome sequencing or taking the DNA mixture from a crime scene. So we will be able to distinguish where are those sequences coming from, either from victim or from the criminals. Or we can distinguish two different molecular subtype of breast cancer, for example, triple negative and luminal A subtype. This is the pipeline. So we have to do the pre-processing of our sequencing data. And then we have this deep learning module here. So we did training, testing, and validation of all the sequencing data. This we replicate many times. So based on using ROC under the curve under area as our evaluation criteria, so we can see the accuracy actually for forensic application is quite good. It's almost 99%. And then for the molecular subtypes of breast cancer prediction, actually we reach 100%. Even though the sample number is still small, but it's very promising from the data I'm just showing here. That's why it was published in a very high impact journal. Another study is for breast cancer recurrence prediction. So everybody knows there's a very famous MammaPrint. It's a 70 gene signature developed by the scientists in Netherlands. So they have a company called Agendia. So you have to take the patient sample and then send it to Holland. It will take 2 to 3 weeks. It will cost $5,000. And then you get this analysis, get the result, and then they will tell you whether you are high-risk patient or low-risk patient. So in addition to doing that, we kind of taking the pathological image, because it takes time and quite costly to do the genetic testing. It costs $5,000 US. So by combining this 70 gene signature, we're taking data from TCGA in the US and then taking their corresponding pathological image and using this deep learning pipeline, CNN, to train to see if we just take the pathological image, we will be able to predict whether you are a high-risk patient and low-risk patient. So we go through many different mathematical algorithms for deep learning, and we find one of them is the best. And actually, this study was just published last month in Frontiers in Oncology. So we will be able to purely just use pathological image to replace this 70 gene signatures. So in conclusions, big data research is, it's very hot fields, everybody, and then it provides a lot of informations. And in order to overcome this big data, a lot of the databases and tools, analytical tool has been developed to help the researcher. And those analysis software, they can provide those researchers without bioinformatics proficiency to do their research much faster and easy by simple clicks and comment. So I demonstrate you 2 examples using deep learning. They can be applied to cancer genomics, and then we might provide an effective therapeutic intervention in the future. So thank you for your attention. If you have any further questions, please send email to me. These are my 2 emails. Thank you.